Hello. Today we will study about the cell division and the mitotic cycle. So what is cell division? Cell division means formation of two daughter nuclei or daughter cell from a fully grown mother cell. That is, formation of two cells is not a segregation or division. It's a division or formation of two cells, that is the daughter cells, from a fully grown mother cell. What are the reasons behind cell division? When cell is metabolically active highly, or the nucleus or the DNA is a doubling of DNA or the nucleic acid present in the cell, or any burn or cut occurs in body, then the cell division will occur. So what are the importance of cell division? The importance of the cell division are mainly repair. Let's see, here I have a cut in my body. To repair that or to fill that part of the body, we should have the cell division to form new cells and to repair them. Regeneration. That is, in some animals like in salamander or lizards, their tails or limbs may cut, and to fill or to form a new tail or new limbs, the cell division occurs and formation of these new body parts happens. Repair, regeneration, growth. Like we all know that every living cells are formed from a first zygote, that is our first cell. From this single cell, our multicellular body, fully grown body forms. So in that case, if cell division will never occur, how can a multicellular body will happen? So that's why growth is an important reason or importance of cell division. So basically, what are the types of cell division? Firstly, this amitosis. A means without, that is just separation or formation of two cells from a single cell. Like in some ciliates or single cellular protozoans, they have this amitosis division system for formation of multiple cells. Second is mitosis. This is known for formation of cell or daughter cell from the same size and same nucleus structure as the mother cell. So this is mitosis. Just like this is a mother cell, formation of two daughter cells with same size and same nuclear DNA present in it. And the last division is meiosis. This is known as reduction division. Formation of cell or the daughter cells from a mother cell having half number of genetic material. Half number of genetic material. Where this meiosis division occurs? In case of germinal cell formation. Just like when a body fully grown zygote forms, it will occurs by the fertilization of one mother cell and a father cell. So when this mother and father cells fused, if the cells are, let's say, 46 chromosome in number, if 246 chromosomes unites, it will form a 92 chromosome structure. So in that case, the living cell will have a abnormality. Okay, so to reduce this problem, we have mitosis division. If mitosis division occurs, the mother cell will form to half nuclei half DNA form cells. Just like here from 46, it will form 23 nucleus, 20 number of chromosomes in the nucleus, and finally ends up with formation of the 46 chromosome zygote. So basically these are the types of cell division. So what are the stages of cell division? Mainly the stages or process of cell division is known as the cell cycle. Why? Because it is a cyclical process. Let's go through with this. So cell division process is divided into two parts. One is interphase. That is the phase between two dividing phase is known as interphase. Another is mitotic phase or dividing phase. Okay. This interphase is divided into another three parts. One is G1, another is S, another is G2. G1 is gap phase 1. S is synthetic phase. And G2 is gap phase 2. Okay. Now, this mitotic phase is again divided into two parts. Why? Mitotic means the division of the cell. Now, by dividing the cell, we have two major parts. One is the nuclear part, another is the cytoplasmic part. That is cytoplasm with another organelle part. So, firstly is karyokinesis. What this two term means? Karyo means nuclear, kinesis means division. Another one is cytokinesis. Cyto means cytoplasm and kinesis means again division. So this is nuclear division and this is cytoplasmic division. Let's study the different phenomena occurs. The first one is G1. In G1, what phenomena occurs? Let's see in G1, all the proteins are synthesized here. So proteins. All the proteins responsible for DNA replication are synthesized in this process. mRNA synthesis occurs and translation occurs in this phase. Okay, then the S phase. S phase means the synthetic phase. Synthetic means the DNA synthesis occurs in this phase. That is the DNA replication occurs in this phase. Okay, that's why it is known as G1 phase. And the third is the G2 phase. In G2, different proteins, different cellular organelles are synthesized. Proteins, cellular organelle, and it's a checking process where the cell is entering into the dividing phase. So this is a checking, and all the organelles that being doubled. Because formation of two cells, so the cell organelle must be also in double. That's why separation of equal number of cells occurs. Okay. Then the kinetic phase. Kinetic phase is divided into two parts. That is the karyokinesis and cytokinesis. It is karyokinesis and that is cytokinesis. Karyokinesis is divided into again prophase, metaphase, anaphase, and telophase. And cytokinesis is an. So this is the main cell division, cell cycle part. Okay, now is a cell division is a major pathway, major process of body cell growth. So for in that case, systematic body must have a checking of this process, because if any malfunction and mal-process occurs and mal-products develop in this process, then it will be fatal for the body and it may end up in cancer. Okay, so for that we have the checkpoints. We have 4 checkpoints here. One is in G1, another is in S, another in G2, another in N. So this is G1 checkpoint. So in G1 checkpoint, when the cells getting problem, then it will end up in a loop formation or G0. Our nerve cells, the non-dividing nerve cells, are entering or residing in G0 phase. So they cannot again transfer into this dividing cells. So our non-dividing nuclear nerve cells cannot be divided. Okay. So here proteins are present is ATR, CH1, ATM, CH2, P53 and MDM2 proteins are present. In S phase, again ATR, ATM, P53 and RPD, DDC1 and MEC proteins are present in the checkpoint. Okay. In G2 checkpoint, that is again ATR, ATM. ATR means CH1 and CH2. ATM means CH2, and topo 2 alpha protein present here. In M checkpoint, here is BUB1, BUB3, okay, MOB, MOD2 and CDC20. These proteins are present here in M checkpoint. Okay, so basically this is the cell cycle and cell division. After this, again two cells are formed and this cell end up again in G1 cycle. So this is a cyclical process. Okay, now in this mitotic cycle we have described that the karyokinesis process is going through 4 substages, that is prophase, metaphase, anaphase and telophase. What? Prophase, metaphase, anaphase and the telophase. Pro means previous, telo means last. Now this is, we will study in a different manner. It is just like a mirror image. Let's study in a mirror image form. Just like in prophase, we have the cells. In the cell, there is a nucleus. Nucleus having 4 nuclear organelles, that is nucleoplasm, nucleolus, nucleoplasm, nucleolus, nuclear reticulum, and nucleochromatin. Okay, now in this process, firstly, from the outer side, nuclear membrane turns to dissolve. Then the nucleoplasm will dissolve. Nucleolus will dissolve, the proteins of nucleus will dissolve, and all the chromatins that are spread up are being condensed to form the chromosome. In the later phase of prophase, it will form the chromosome. That is the prometaphase. In the prometaphase, between the metaphase and prophase, in the prometaphase, that will form a chromosome structure. Okay, there is a centriole organelle just lie above the nucleus. It has 2 centrioles. The 2 centrioles are going in a 90-degree fashion into the end of the 2 nuclear, the nucleus, 2 pole of the nucleus. Okay, in the metaphase, the centrioles are arranged in the 2 pole, and all the chromosomes, the form, that is the condensation formed by the condensation of nuclear chromatin, they are arranged in the middle or the nuclear region of the cell, the middle of the cell, equatorial region. It is known as the equatorial region. Equatorial region of the cell. Okay, now up to this phenomenon occurs in the anaphase. Anaphase will be reverse of the metaphase. Now in the metaphase, all these chromosomes are arranged in the middle or equatorial region, and astral tubes are from these centrioles. It will attach with the kinetopo region of the chromosome. Okay, in the middle of the kinetopo. This is the point of region in the middle of the. In the anaphase, reverse of this occurs. These chromosomes will move from equatorial to the terminal position. Okay, and these astral tubes will be become short by depolymerization of the proteins to form these astral tubes. Okay. In the telophase, it will reverse the prophase. Like in this phase, the separation of the chromosomes will end up. Chromosomes will form nucleochromatin, nucleolus will reappear, nucleoplasm will reappear, and eventually nucleomembrane will reappear to form two nuclei cell. Okay. Up to this, karyokinesis occurs. Then the cytokinesis. In animal cell, cytokinesis occurs by just formation of furrow and separation of two cells. But in plant cell, presence of the cell wall is very much crucial. So this furrow cannot be occurred. In that case, plant cell have a phragmoplast. So this phragmoplast just this phragmoplast will stores in the middle or equatorial region of the cell, and by this two nuclei cells will be separated. This is known as the cell plate formation. Okay. Now this is the main process of cell division. In Fleming, scientist Fleming in 1880 first observed cell division in salamander. So basically these are topics. Now let's have an MCQ. Like if in question we will ask that in which stage of cell division the DNA replication will occur or DNA duplication will occur, it will be in the S phase. Now if someone asks that in cancer which cell division is appeared and which cell division occurs, so what will be the answer? It will be the mitosis. In cancer, the rapid division and formation of same type of cell is essential. So in cancer, the mitosis is a cell division. So basically, thank you. Okay.